Light detection is an ancient sense, known in every major group of living organisms, including bacteria. Distinguishing between different wavelengths of light, that is, color vision, is even demonstrated in the wavelength-dependent phototactic responses of certain bacteria. Most organisms rely on proteins known as OPSINs for the detection of light, and the rhodopsin group is one of the largest families of the G protein-coupled receptor superfamily. Members of the rhodopsin family have been modified to perceive a variety of substances in addition to light, such as biogenic amines, such as dopamine, short peptides, proteins, nucleotides, lipids, eicosanoids, and other substances. Bacteria rhodopsin, the rhodopsin of bacteria, is homologous to eukaryotic GPCRs, although the order of the transmembrane helices is different. Homologs of this bacterial protein is known in primitive eukaryotes as well. In archaea, the archaebacteria, opsins also bind to the retinol protein, just as in vertebrates, and this opsin functions in phototaxis and light-induced ion transport. The eye is made of neural tissue, and technically the retina is part of the brain. It forms as part of the brain during embryonic development. Therefore, the optic nerve is not technically a nerve, but rather a tract which connects the retina, which is one part of the brain, to the rest of the brain. One can observe this in embryos where the optic cup, which will form the retina, actually forms as an out pocket of the embryonic brain. There are a variety of opsin proteins which are light detecting and which are expressed in the brains of animals, especially more primitive vertebrates. Thus it seems that the vertebrate eye just developed as a special part of an already light-sensing brain, a brain which expressed these light-sensing opsins and then specialized one part of the brain to move forward and function as eyes. Even before vertebrates evolved, the chordate lineage had duplicated opsin genes, and 3 opsins are known in tunicates and as many as 6 in lancelets. Humans have at least 8 opsins in their genome, which is about the same as most jawed vertebrates. Vertebrates not only possess photoreceptor molecules in their eyes and their retinas, but they can also possess opsins in other retinal cells, those not dedicated to vision, in their deep brain, in their pineal complex, and even in the skin. In non-mammals, the iris and melanocytes of the skin can mediate responses to light. All non-mammals have photoreceptors in both their deep brains and their pineal glands, at least during embryonic development. In halibut fish, for example, the photoreceptors of the pineal gland, rather than those of the eye, are capable of photodetection before hatching. Different groups of vertebrates have had different duplications of opsin genes, allowing for a variety of differences in color perception. For example, some fish and birds can see an ultraviolet light because of an opsin which has been modified for detecting that type of light. Not only can higher primates detect the difference between red and green, but so can birds, although this has arisen from a different opsin duplication than that responsible for red-green vision in primates. And so different animals can have different opsin duplications which contribute to color vision.